for 2 years, and he is now a visiting professor at the UK, Germany, for 18 months. He had 47 articles published in high-profile international journals, and he is also a member of the Scholar Welfare Office at Assiut University. His speech today, with the title of Genome Mapping, reveals the effect of genome by environment. You have the mic.
Okay. Thank you, Dr. Muhammad, for the nice introduction. And today I'm going to give a talk about, or it's a very important talk about GxE interaction and how can we use the molecular genetics tool to overcome this problem to identify the highest yielding genotypes. So as Dr. Muhammad said, this is a title of presentation, Genome-wide association mapping reveal effect of genotype environment interaction for. This is the contents of my presentation. I will start with a small introduction about problems of genotype and environment interaction, and then I am going to speak about molecular breeding program to select the promising genotypes that we proposed, and then I will talk about effect of genotype environment interaction on green yield as an example. And I will end my presentation with molecular approaches to overcome genotype environment interaction. On to the introduction. So all of us, I will just go quickly for the introduction. All of you know that wheat is very important crop because all of us eat bread, not only in Egypt, around the world, and it occupied after maize and rice. As the population around the world are growing, the breeders have focused on the development of cultivar having higher yield in combination of biotic and abiotic stress tolerance. We have two main issues for improving green yield. The first one, multi-environment yield trials. So we have to test wheat genotypes in more than one location and in more than year. Why? To identify superior genotypes in plant breeding programs and to determine where the cultivar are best adapted. Genotype environment interaction is a main problem because this pointer. Okay, sorry. Because it reduces association between genotype and phenotype. Second, reduce the heritability. The second issue is grain yield is controlled by many genes. It's polygenic traits, which means it's controlled by so many genes, not single gene or two genes, so many genes. These genes have major effects, and some of gene has minor effects. So actually, to genetically improve green yield, we have to identify as many as genes for improving genetic improvement of green yield. Traditional breeding program, you know, it takes a long time and is very labor and time consuming. You have to cross to plant F1, F2, F3, and go to F7 to have highly homozygous genotypes. But the advances or the current advances in DNA technology provide very new methods for sequences, which can provide a lot of markers, like single nucleotide polymorphism. And such huge number of markers can be used by many genetic analysis associations, such as genome-wide association. This is a very important method which allows us to identify allele and genes associated with target traits. Could be green yield, could be disease resistance, could be tolerance to drought or frost tolerance, as an example. Also, it allows us to go further genetic analysis, such as genetic diversity and identifying number of target alleles. So here in this talk, we used the combination of all genetic analysis, like genome-wide association mapping, we incorporate genetic diversity, we incorporate the number of genes in each genotype. So the main objectives of our research is to study first the genetic variation and genotype environment interaction for green yield in a set of 270 genotypes, which is very large number. And these genotypes were grown in different environments. The second objective, to identify the highest yielding genotypes at different environments. And the third one, to use molecular breeding approaches to overcome the genotype environment interaction. So this is the scheme of molecular breeding program that we suggested. So first we have to start with big population of genotypes. This is, we should use diverse collection. So these genotypes, we do it should be like phenotyped, okay, and genotyped in the same time on the same period. So the purpose of phenotyping is to select, to have a selection. So the first step is to select for the target trait. So if you are going to like evaluate green yield, so you select for high yielding or to most resistant genotypes for like stress or striblast, or high tolerance genotype for drought or heat stress. In the same time, the same population should go for genome-wide association study. So you do sequencing and you have markers, and then you combine the marker and the phenotypic data to identify genes associated with your target trait. And after the step 2 and step 1, you have like very few genotypes that performed very good for high yielding or high resistance to we use the advances in genome-wide association to identify how many genes in each selected genotype. So for instance, here you selected 6 genotypes. It is high yielding or high resistance. And you already identified number of genes in all population, 270 population. Then you go for in each genotype, genome of each genotype, and you how many significant SNPs in each genotype. So for instance, the first one is 13 SNP markers, significant SNP marker associated with target trait, and we have like here 7 and here 5, here and here 13. And then the third step, you should, yeah, this is how I told it, to identify number of significant marker in each step. Number 4 is to study the genetic diversity among the selected genotypes. Of course, genetic diversity is very important. So you have to measure how your genotypes are divergent. Okay, and you know from dendrogram analysis. Okay, and the first number, number 5, the step number is to identify number of different target allele among each pair of selected genotypes. Okay, here for instance, if you go to this figures of the selected parents, you have here like 25 SNP in this genotype and 20 SNP in this genotypes. You may find that 20 genes are similar in this parent and this parent. So we have to look for how each pair of selected genotypes are different in the number of genes. And then many genotypes we have 14 genes. From these genotypes we have 20 different genes, because this is very important for crossing. And then you come with final selection crossing to produce promising cultivar with target traits. Of course, if you have high performance genotypes for high yielding and high genetic diversity, and here we have like 20 genotypes, 20 SNPs, significant SNPs associated with high yielding, and the second genotypes also has the same 20 SNPs associated with high yielding. So when you do crossing, you will not have improving, because the same genes here are in the first genotype is the same genes in the second genotypes. So it's better to look for how the number of different genes between each pair of selected genotypes. So come to the effect of genotype environment interaction. This is the population that we used. As you see here, we have F3, some from third generation and some of sixth generation. But keep in your mind that in each genotype that we use came from two different crossing between two different parents. So we expected that the all 27 genotypes are highly diverse. And this is just illustration about the production scheme of the population. So we will apply the protocol that I suggested. We phenotype green yield in 9 locations, which is many locations, and this locations in 2 states. This study was done in US during my postdoc, and I continued after I came from USA. So we published this last year, and we grow these genotypes in Nebraska state, and in Nebraska state we have 8 locations, 8 cities. As you see here, the red points. This is like some from south, extreme south, some extreme west, extreme east, and this is like Nebraska and the Kansas State University. So we aimed to planted our genotypes in different cities or parts which are far from each other. And we have to go for ANOVA, analysis of variance, and as we expected, high differences among environments we found, and of course we have to look for the interaction between genotype and environment, and it is very high significant differences. So in this step, when we go for genetic analysis, so we can to combine all the 9 environments together. So we have to perform the genetic analysis in each environment. Once you have significant differences between genotype and environment, we have to perform genetic analysis in each environment. This is correlation matrix between in green yield among the 9 location. As you see here, when it becomes dark, it's more significant. So we don't have high figures of or how high numbers of correlation. Maybe the highest correlation between Grant and Sydney, this so McCook, and between Mead and Lincoln. But otherwise, as you see here, very weak significant correlation in green yield among the environments. This is also GG plot. As Mead and Lincoln, we have McCook and Grant here, far from each others. So this is good evidence that the climate change and the weather affect green yield. So we have only very few group here close together, but we have here Mead and Lincoln very far, and McCook and Grant are closely related, but it is very far from the other groups. To expand our understanding about the effect of weather and the climate of regions environment, we analyzed high temperature and low temperature data, and as you see, we thought that is like all environment have a stable, a little bit stable, or the similar trend in high temperature and low temperature. But if you look for the rainfall and the snowfall, as you see, in each month you have ups and downs in each location. And we thought that rainfall and snowfall in USA was the main factors for affecting the green yield of wheat. We also performed principal component analysis. So here the red point is the temperature. Temperature also difference between among the 9 environments. Rainfall also is different. Snowfall here, as you see, North Plate is separated from others. So it was very important to understand the climate and the weather in each location to interpret our data. So come to our scheme, molecular building scheme. So we apply the scheme which I described it to our study. So first we phenotypic selection. Here we in each location we selected the highest 50 genotypes. Okay, we sorted all genotypes from highest to lowest, and we selected 50 genotypes. I mean, you can use 20, you can use 30, based on your objectives and based on your number of genotypes that you have. And then after we selected 50 genotypes in each location, we looked for the common genotypes across all locations. And we came up with this table. We have 13 wheat genotypes. For instance, the first one was among the highest 50 genotypes in 8 locations, as you see here. And we have some genotypes were among the highest 50 genotypes in 4 locations. And please keep this in your mind, 25 and 26 genotypes, because from the phenotypic data, oh, okay, good. They are among the highest 50 genotypes in 8 locations. But you will change your mind in the next slides. And we go for genotyping. We extract DNA, we send it for sequencing, and this is like genotype by sequencing methods. Don't have time to describe this, but from this method, we have got a lot of SNP marker from this population. We perform genome-wide association study. We got from GWAS 11,091 SNP marker, and we perform genome-wide association mapping. This study I published with Dr. Shams. He will speak us. And we have a nice study about genetic diversity among this population. We published in Frontiers in Genetics. This is genome-wide association table, and this is the number. The first or second column is the number of SNP markers that we found in each environment. Sydney and North Plate and Lincoln, we found like 11 SNP markers in this location associated with green yield. And also we have a Grant. Grant is only 3 SNP marker, so it differs based on the location. The number of SNP marker associated with high yielding, which show significant association, differs between among environment. Of course, as we expected, we didn't find any common SNPs among the environment. So each environment had its own significant SNPs. And most of people, when they found no significant marker, they don't need to publish the data. So because most of breeder and geneticists like to have, we have significant marker in 3 locations. But here we have like different concepts. And this is, I would like to show the p-value of significant marker. It is like a very, very low. This is Manhattan plot to show the significant marker in each location. It is far from the p-value of other markers. Also, it was very interesting to know how many SNPs located in each chromosome. So we found in chromosome 2D, this chromosome 2D, we have 13 significant SNPs. Of course, 13 significant SNPs across all locations. So we go further for linkage disequilibrium analysis to know if this 13 SNP marker co-inherited together to next generation, or there is no relation between these 13 SNPs, significant steps. And this is the LED. The red colors, or when it becomes more red, it means significant LED or high LED. So this group of marker is very high, or have high LED. So it means that they co-inherited together from generation to generation. Okay, and we found this marker from mid location, our environment. We also have these markers from Sydney and this marker from Gap. This is the 13 markers, 13 significant SNP markers that we found. So this is because G by E. So each significant SNP marker we found is specific for location, for environment. We looked for where these significant markers were located, or in genes of wheat genome. We found this group, which is high LED, where we found they are located within this gene, and this gene includes two proteins, zinc transporter and metal, ion transporter, and this protein has high association with increasing green yield. So this is one of the advantages of using genome-wide association mapping. So you found gene associated with really target traits. This group of high significant LED of marker also we found well located within two genes model, which included two transporter and calcium exchange. Also, this protein has a good function to increase green yield. And this results indicate that our genome-wide association analysis was successful because we found the significant SNPs has a good association with green. Step 3. We have identified 13 genotypes and we have identified genes. Let us to go deep into the genome of each genotype, of each genotype. So this genotype has like 43 significant allele or significant SNP marker across all locations, and so on, as you see here. So this is 35 genotype. We have like 46 significant marker and 50 0. This is had the lowest number of significant marker, maybe like less than 40 significant marker. So this is very important information. Now I have identified superior genotypes phenotypically, and I know now in each genotype how many genes in each genotype that can increase the green yield. Step 4, genetic diversity. This is the genetic diversity matrix. It gives very important information about the distance between each selected genotypes. So here we have this 25 genotype number 25 and 26, I think, 26. It is 0.0, so high genetic distance between these two genotypes. Just keep in mind that I marked these two genotypes in the phenotypic selection table, okay, because these two genotypes were among the highest 50 genotypes in 8 environments. Also, they are highly divergent in terms of genetics. The highest, lowest genetic diversity were among 24, 624, and 661. This is the lowest genetic diversity we have. Step number 5, number of different favorable alleles. So we have 13 genotypes. So we did a matrix. So see here, the two genotypes which I marked in the phenotypic in the selection table, only 9 different genes between these two genotypes. If we look to this matrix table, you find 479 genotype and 533. There is 32 different genes between these two genotypes. So if you cross between 25 and 26, this line, you will have, of course, yeah, maybe better F1, but if you cross the between 479 and the genotype number 533, the F1 will get 32 different genes. So this is also very important information, because this is, by this way you will shorten the breeding cycle. So by how you select the promising genotypes. Come to the conclusion. Genotype environment interaction needs to be investigated for identifying genotypes that can be used for improving green yield in all tested environment. Utilizing the advances in DNA sequencing and genetic analysis by genome-wide association mapping has helped in identifying possible candidate genotypes to be used in improving green yield in a group of environment. 3 criteria were suggested to identify candidate genotypes to be used as a parents for crossing in a group of target traits. These 3 criteria depend on combining the information at the phenotypic and genotypic levels. This research provides a framework of considering how to select parents for future crossing. And this study was published in PMC Genomics. This paper, and within this paper you have really many details about good crosses and bad crosses among the selected parents. So we provided many examples in these papers how really to overcome the selection of the problem of genotype environment interaction. And this is very important because you, for instance, you have crosses that can fit with like 5 environments or 3 environments. Instead of you design breeding program for each specific environment. Okay, so just I would like to go this slide. So here, what is the best crosses here? The best crosses is 625 and 4779. Why? Because the two candidate genotypes have 40 target alleles, different target alleles, among the highest 50 genotypes in 5 environments, and they have like 30 different genes in each genotype. So by this way, you can confidently go for crossing and for future breeding program. And we used the same protocol in paper we published in Frontiers in Genetics in stem loss resistance. Also, Dr. Shams will give a talk about this paper in after lunch. And of course, I would like to thank Dr. Stephen Banziger, the University of Nebraska Lincoln, USA, my nice colleague and the kind colleague, Dr. Shams Tahir from Sadat City, and Dr. Ahmed Sallam from Qatar University. And thank you so much for attention.
Thank you very much, Dr. Ahmed, for your for all this movement for question.
This one? No, this is different in steps. Yeah. No, gene. Yeah. It's, yeah, could be maybe, yeah, could be 3 alleles in a gene. Yeah. This is good question, Dr. Helmi. We didn't investigate this. I mean, you are correct. So maybe like, okay, maybe the 32 alleles in 15 genes, in 15 different genes. Yeah, we good point, but we didn't investigate. But we expect because you know this is 32 different alleles, maybe significant. Maybe they are significant in like more than one locations. So I believe this is each SNP is like maybe one gene or located or maybe two SNPs in one gene or something like that based on but we didn't investigate the high LD. Yeah. Yes. Good.
خلاص يعني كده تمام. خلاص ماشي كده بسكمامة.
Yes. No, for each location. For each location.
ياسمين بتقول ياسمين ايه؟ جمانة بتقول لي لو قابلت ياسمين هناخد عيال وحش.
Highest way. Yeah. Green yield per plot. Yeah, it's for plots. Yeah. Yeah, plot 1 meter square. Yeah. No, it is like the population originated from Nebraska breeding program, and then we got this seeds and we distributed in a cross location. All the crosses, the scheme of production that I described it were done in Nebraska, and we propagated in Lincoln, exactly because I was working in Lincoln University, and then after propagation we planted in different environments. Yes, but statistically you don't have correlation. Yeah, in green yield among the 9 genotypes. Yeah. Thanks. Stop recording. Thank you, Dr. Muhammad. Thank you.